So let's talk now a little bit more detail about how mRNA is processed in eukaryotes. There's going to be three main ways that this RNA can be modified. And the first thing that happens is there is a cap that is put onto the 5' end. The primary goal of this is to help a ribosome determine where to attach when it's ready for translation and out in the cytoplasm. Another thing that will happen is that there is going to be a poly A tail put onto the 3' end of the transcript. This is going to have a couple of different purposes. First of all, it is going to help facilitate the transport of mRNA out of the nucleus, and it is also going to help inhibit degradation of mRNA. RNA in general is very, very easy to degrade. There's enzymes all over the place that can degrade it. So by having a nice cap on the end like this, it will help that RNA and protect it from degradation. The third processing event that will happen will be splicing. So mRNA, especially pre-mRNA, is composed of exons and introns. These are just different regions of the gene. The exons are going to be regions of the gene that will be expressed in the final transcript. The introns are going to occur in between the exons, and they are not going to be expressed. This has several different benefits to it. It can allow a cell, first of all, to pick and choose which exons will go into a particular mRNA. And it can give the potential gene many different types of potential proteins that it can make. After these three processing events, the 5' cap, the 3' tail, and the splicing, we now have mature mRNA. And finally now, at this point, is the RNA ready to leave the nucleus and proceed with translation? And we're going to talk more about that aspect next week. I just want to point out, in prokaryotes, these introns can be removed. They usually don't have them, but they can have them. If they do have them, they are removed by self-splicing. So they don't have enzymes for removing them, but in fact the intron itself has the capability of enzymatically splicing itself out of the pre-mRNA. So why might a cell have these introns? Well, if you look at the number of different genes that are possible, compare that to the number of different transcripts that we get, what we see is that as we get more and more complex organisms, you don't just simply have more and more DNA. The number of protein coding genes just doesn't keep pace with how complex different organisms are. However, the proportion of the genome that we have that is introns does increase. So in humans, our genome has about 20 to 25,000 coding genes, and that number is actually not 100% known at the moment. It's probably a little bit closer to 20,000 though. Up to 95% of the DNA in our genes is actually introns. They used to call this junk DNA. They thought it was just useless. We now know that that's not exactly the case. Although it may not be transcribed, it may also have an important role. So some of the important or possible functions of these introns, well, first of all, these exons can combine in various combinations, and we do know that this occurs. So if you have, say, exon 1 from one transcript, you can combine that with exon 3, and I'll show you what I mean in just the next slide. And what this means, though, is that different mRNAs can result from one segment of DNA. And this is indeed the case. We see this with many different genes. Some other possible functions are that introns may regulate gene expression. They may also be regions to encourage crossing over during meiosis, so helping increase genetic complexity. So taken together, there is an exciting new picture of the genome emerging, and trying to figure out, now that we know all of the sequence, how is it being transcribed? That's really the most important part. So let's look here at some of these introns and exons. So again, introns are going to be the regions that are not going to be expressed. In this particular DNA template, you can see here exon 1, exon 2, exon 3. In your pre-mRNA, you've got some regions here, okay, a leader sequence, you've got some exons, you've got some introns in blue. The orange is the part that the cell is going to keep. After processing, you're going to add your 7', the methyl G cap onto the 5' end, the 7-methyl G cap. Onto the 3' end, you're going to have a poly A tail. What I mean by this poly A tail is literally a whole sequence of adenines. Okay, A. So after the cap and the tail are added, you have splicing. In this case, these exons are going to be removed, added together, okay, and now you're going to have your mature RNA. And as you can see, these intronic regions, these are going to be removed. Not sure why they don't have the poly A tail and the methyl G cap on here. I know part of it is removed. However, the poly A tail is still present typically even when it's in the cytoplasm. So here's some more examples of some different genes. Mouse insulin, you can see, only happens to have one intron. It's fairly large. Rabbit beta globulin has two. And now you can see something like chicken ovalbumin, and it has a whole number of different introns that can be removed. And relatively very few of that gene is going to be retained as exons. Here's some more table contrasting gene size versus the mRNA size with the number of introns. If we look at something like insulin, it's relatively small. It only has 2 introns. If we look at some genes that are a little bit larger, okay, maybe potentially a little bit more complex, you can see the number of introns increasing quite dramatically in some of these cases.